So in our previous units, we've looked at how chromosomes coil up in order for a cell to reproduce. But now we're actually going to work our way backwards. We're going to uncoil that chromosome, okay, all the way back down and take a look at the structure of DNA and figure out exactly how does it control the inner workings of a cell? How does it determine exactly what traits an organism gets and which ones it doesn't? So, DNA is named so from the word deoxyribonucleic acid. It's one of the nucleic acid organic compounds, and it is the molecule that ultimately controls the production of proteins in the cell, typically referred to as the blueprint for the cell. Now, DNA has two basic jobs. One, it stores information, and two, it copies itself for new cells. So there's two basic jobs there. DNA is considered a polymer. It is a larger macromolecule. It is composed of smaller subunits or monomers called nucleotides. The nucleotide itself has three basic parts: deoxyribose sugar, a phosphate group, and what we call a nitrogen base or nitrogenous base. Okay? So let's take a look at this, because how is each of those nucleotides different then if they all have the same three things? Well, the nitrogenous base could be any one of four different types of bases. It could be adenine, usually represented by an A, guanine represented by a G, thymine represented by a T, and cytosine represented by a C. And what I need you to do right now is right there where that C is, change that to an S as I'm looking at this. That's wrong. Make sure that's an S right there. So a complete molecule of DNA consists of two linked strands together. So DNA gets put together like a ladder. The sides of the ladder are sugars and phosphates, while the rungs are those nitrogen base pairs. So I say base pairs because that's what makes up the rungs of the ladder, the part that you'd literally step on. Okay? The bases get paired because adenine and thymine fit together, and cytosine and guanine fit together. Now, the reason they fit together is because they're just like puzzle pieces. Their chemical structure is configured in such a way that they fit together like puzzle pieces. An adenine would never fit with a cytosine. It doesn't fit with a guanine. Okay, just like a guanine wouldn't fit with a thymine. So when we look at them, it's because they're categorized into two basic groupings. So there is the purine category, which contains adenine and guanine. Then there's the pyrimidine category, cytosine and thymine. When you look at the green, the red, the purple, and the yellow structures, what is it that makes them different? Why are they separated into two basic chemical groups? Hopefully, you recognize that the purines have two rings of carbons. That's what's represented by those shapes, where the pyrimidines only have one ring of carbons each. That's what makes them fit together. Imagine if cytosine and thymine paired together. That would be a pretty small rung. And if guanine and adenine fit together, that would make a pretty large rung. Have you ever climbed a ladder that had wavy outsides? Probably not. So the base pairs are held together by really weak hydrogen bonds. If you remember, hydrogen only has one electron, so that's why it forms really weak bonds. It's very, very easily broken. Then those two strands get twisted like a double helix, and that's what creates the twisted ladder shape of DNA. So we've discovered DNA some time ago, and it was first described by two scientists named Watson and Crick after viewing a picture taken by Rosalind Franklin. She used X-ray crystallography in order to take the picture that you see on the right-hand side of the screen of DNA, looking at it, looking down, as if you were standing at the top of the spiral staircase and looking down. So the information stored in that molecule of DNA is coded by the sequence of those nitrogen bases. So here, we're looking at the structure of the DNA double helix. So notice how, looking at a nucleotide, and this is actually what I want you to do. Looking at a nucleotide, there's the sugar, there's a phosphate, and there's a base. If you flip to the back side of the page that you were just taking notes on, there is a ladder-like structure, and there is a double helix type structure. Do me a favor and take a look at the ladder structure, not the twisted one, but just the ladder type structure. And notice how there is a base, okay? For example, let's find a cytosine. So on the left-hand side of your structure, there is a cytosine, okay? Then next to that, the cytosine is actually attached to one of these sugars right here. And on your diagram, it's not labeled, but would you go ahead and take and label that with a D? That sugar right there that the base is attached to is the deoxyribose. And then on your diagram, the phosphate is right above it. It's in between each of the deoxyriboses. So go ahead and circle a nucleotide and label it as a nucleotide there. So that way you understand that that's the repeating structure that makes up DNA. All right, so you can see how the structure ends up getting twisted. It actually literally looks like two separate spiral staircases twisting up. It looks pretty neat. Okay, but when we look at DNA, oops, sorry, I didn't switch that. When we look at DNA, the other role of DNA is that it has to replicate itself. So there's what's called self-duplication of DNA. And the DNA is going to unwind because it has a hard time when it's all twisted up like that, replicating itself. Then helicase comes through and unzips the molecule by breaking hydrogen bonds. Each strand then acts like a template, and DNA polymerase matches up the free nucleotides, brings in free nucleotides that are already floating in the cytoplasm. So polymerase and helicase are both enzymes, and hopefully you recognize that because each word ends in ASE. Okay, so right here ASE and right here ASE. That's how we know that they're enzymes. Okay, so as we keep going, new hydrogen bonds form then in order to connect each nucleotide together as well as the ladder together. The DNA polymerase then links the strands together, forming those hydrogen bonds between the bases, and the DNA can rewind back to form that double helix, that helical type structure. Now, I was just going to say something about those helicases. Here's what I want you to think of. And actually, let's do it this way. Okay. Number 2, this step right here, okay, this helicase reminds me of scissors. So I'm going to write that down. Not very good handwriting. I know I'm writing with a mouse, okay? But a helicase is a lot like a pair of scissors, and it goes straight up the middle of that ladder, just cutting it. The DNA polymerase acts a little bit like a helper, okay? It's what actually goes in and makes the new molecule of DNA, all right? Now, where's my thing so that I can get rid of the pen? Oh no, now I'm writing all over the place. Ah! So the cell has enzymes then that will actually go through. See, it's not letting me click forward. Oh no. Pointer options. Let's get rid of that pen. There we go. And it's not getting rid of the pen. And now I just ended the show, which I did not mean to do. So what you needed to see at the very, very end, oh my goodness, sorry about that. What you needed to see at the very end was that the cell has enzymes that proofread and repair the DNA. So it's looking over to make sure that the A's and the T's pair together, the C's and the G's pair together, to make sure that there's no errors or mutations. And if they come across a pair that is not matched up correctly, or there's a G in place of a C, then the DNA gets repaired. So that's the really cool part of it. So here's what you need to understand is the whole DNA helix does not actually unwind. And you don't have this diagram in your packet, but only sections of it actually unwind. And that's what we call the replication fork. So these two sides split apart, and then that DNA polymerase, which is represented by the kind of turquoise green ball there, is going through and pairing up the bases and will work its way all the way to the end of the replication fork. So it's kind of interesting that way. I'd like to be able to show this video to you about DNA replication. So let's take a look. Oh, media not found. No way. That's too bad. I wonder why it's not working. Oh well, if I get it to work, we'll watch it in class. Cool? Here's something that's interesting. The percentage of bases found in 4 different organisms. So when we look at a type of bacteria called Streptococcus, you may have had strep throat before, look at the combination of base pairs. Notice how A and T are supposed to go together. They are approximately the same, but not exactly the same in percentage found in the DNA. Likewise, G and C pair together, but still they're not exactly the same percentage. Interesting, huh? Look at humans. Pretty dang close. Pretty close. Why do you suppose there's more variation in the bacteria than there is in the humans? Well, it could be because bacteria mutate and adapt so quickly compared